Welcome to the Whole Health Podcast, where we discuss science-backed information to help you optimize your health. I'm Jonathan Jarecki, and I'm a pre-med undergrad student passionate about health optimization and preventative medicine. My guest today is Dr. Gerald Pollack. Dr. Gerald Pollack is a professor of bioengineering at the University of Washington, where he leads an active laboratory best known for the work with water. His lab is credited with discovering and characterizing what's known as the fourth phase of water, or exclusion zone, EZ water, a form of water that exists in a more ordered, structured state when exposed to radiant energy or light. Humans are made of roughly 2/3 water by volume, but more strikingly, over 99% of our molecules are water molecules. Dr. Pollack argues that the water inside our cells isn't just passive and liquid water, but most of it exists in this structured EZ form. This has major implications for everything from cellular function to bioenergetics, and it challenges the foundations of how we understand biology, chemistry, and physics. As Dr. Pollack has said, the current scientific theories need a change, and his lab is at the forefront of that transformation. These are the kinds of ideas that could shake the earth and spark revolutions in science. In our conversation today, Dr. Pollack and I delve into the science behind EZ water, its relevance to human health and biology, and what it can mean for the future of medicine. If his findings hold true, they may warrant the rewriting of biology textbooks as we know them. Dr. Pollack is also the author of several acclaimed books, including Cells, Gels, and the Engines of Life, and The Fourth Phase of Water, both of which are linked in the show notes. If you're interested in learning more about his research or supporting his lab, I've also included a link to his laboratory's website. You'll find that the show notes are extensively written for your convenience. Before we begin, a quick thank you to today's sponsor, Biojit. Biojit provides premium, third-party lab-tested Shilajit. For those who don't know, Shilajit is a potent natural resin formed over thousands of years as plant matter and minerals decompose in the mountains. This ancient substance emerges from rocks and has been used in Ayurvedic medicine for over 3,000 years. Shilajit is packed with minerals and bioactive compounds and is renowned for its wide-ranging benefits, including, but not limited to, hormonal balance, increased testosterone, enhanced libido, improved mitochondrial and bone health, reduced inflammation, and a greater overall energy. I've researched numerous Shilajit brands, and Biojit certainly stands out. Each batch is hand-collected, filtered, and purified under pristine conditions, a process they have kindly allowed me to see for myself. Most importantly, it's rigorously tested in the US-based third-party labs to ensure purity and potency. If you'd like to try Biojit, click the link in the show notes and use code JonathanJ, that is JONATHANJ, at checkout for 10% off your order. Again, that is JonathanJ at checkout. And now for my discussion with Dr. Gerald Pollack. Dr. Gerald Pollack, welcome.
A pleasure to be here.
Awesome. So today we're going to be talking a lot about a bunch of different topics. We're going to dive deep into the science and see and hear about all your research, and I'm super excited for that. But before we get into all that, I would love to hear where your love for science came from.
Oh, well, nobody ever asked me that question, and with a love for science. Well, I think it was from a few colleagues of mine who informed me that just because something is in a textbook, it doesn't necessarily mean it's right. And boy, I absorbed that message. And at the beginning of my career, I was not studying water. I was studying muscles and how muscles contract at the molecular level. And there was a guy who was, you might say, the most distinguished and important scientist in the field. And his name was Sir Andrew Huxley. And he was a member of the famous Huxley family, master of Trinity College, Cambridge, president of the Royal Society, Nobel Prize, every distinction that you can imagine. And here was my colleague, originally from Japan. His name was Iwazumi. And he came, I was in graduate school at the time, and we started working together. And And he told me that Huxley's theory is wrong. And, you know, I looked askance at him and said, well, how could it be? Because this guy is so distinguished and so famous. He said, well, I'll show you. I'll explain to you why he's wrong. And it took 5 minutes, and he convinced me that that theory couldn't possibly be correct because it violates some fundamental principles, simple principles, not complicated principles, principles of stability. You know, and everything needs to be stable, otherwise it would fly apart. I was astonished because such a great man, you know, distinguished Nobel laureate, how is it possible that he could be wrong? Well, it was apparent that he was wrong, and that started me off on a career. Really, it was partially launched by this Japanese colleague of mine, and I learned from that, you know, that just because something is in the textbook, it doesn't mean at all that it's right. It may be right, but it may not be right. Yeah. And it may not be right because it violates principles of logic, or other fundamental principles. Yeah. So Sir Andrew Huxley became my, you might say, friend, colleague. He passed about 7 or 8 years ago. And every time I saw him at a meeting, or at a conference, I avoided him because, I didn't really have a lot to say to him. You know, and what do you do? Is that nice weather out there, or whatever? But that's where, I mean, your question, how did I get involved in science? You know, it's pretty compelling when you realize that you can contribute something to science, something fundamental, even something that's actually widely accepted, maybe totally wrong. That's exciting. Yeah. And that started me off. And I met other people like that who were equally impacting in my career. Yeah. Does that answer your question?
It does, yeah. Could we go a little deeper and actually go a little further back in time? Like, so growing up, in high school, were you determined to be a scientist, to do research?
No, never. I didn't know what I wanted to do.
Really?
In
okay.
High school.
And so was it college that?
Well, I studied electrical engineering, and you know, that's a technological thing, and there aren't too many questions that you can raise. But it turned out, in fact, actually, there are questions. It was a pretty thorough grounding in fundamental principles, but never did I think that I, you know, would become a scientist. And then I noticed there was a program in biomedical engineering, and this was actually the first program in the country. It was at the University of Pennsylvania. So I thought I'd apply. And I remember my mother encouraged me, and the reason she encouraged me, I think, was not because she was interested in biomedical engineering. She had very little education, but I had a girlfriend, and my mother didn't really care for the girlfriend, and she thought, oh, well, if he moves from New York to Philadelphia, the girlfriend will be obsolete. And that's what happened. So, you know, and even that program in itself was not really so inspiring. If I remember back, you know, I'm studying water now, and the chair of the department was also studying water, but his approach was really boring, and I had no interest whatsoever. Never did I think I would actually go into the subject of water. It happened 20, 25 years later. So no, I didn't start out that way at all. It was only after a year or two in graduate school when this Japanese guy showed up, and he started telling me, just because something is in the textbook doesn't mean it's right.
Yeah, that's so awesome that you were exposed to that idea early on, because I mean, my whole philosophy is question everything.
Absolutely. Yeah. Question everything.
Exactly.
You're on the right track.
Exactly. It's in my Instagram bio. I tell it to everyone. If people ask me, do you have one thing to tell the world? It is to question everything.
Oh,
because
you.
And you write a really awesome quote. I believe it's in Cells, Gels, and the Engines of Life. You say, assumptions are weeds that choke the flowers of truth. It's
I
so awesome.
I think it probably wasn't original. I probably got it from somewhere else.
Yeah.
But I don't remember.
Yeah, it's a great quote, and it's so true. You know, these assumptions that we make that everything in the textbook is correct can bring us down, or can lead us to studying stuff that is just fundamentally wrong, right? If you build a foundation on sand, well, that's not a really great foundation. Right?
No, it's not.
And so that's that.
But you know, Jonathan, you know how it gets there. Someone in science comes up with an idea, really fundamental idea, and shows to a few colleagues, a few influential colleagues, and those colleagues say, well, you know, this is pretty interesting, pretty good, and then the person writes a few papers, and he gets a few accolades, and pretty soon he's invited to write a textbook, and he decides, well, I think I'll take the time to do it. And the textbook is published, and before long other people write other textbooks of similar nature, and the publishers love it. They don't want to, if they have a choice between publishing something that is pretty well accepted by a few experts in the field, they rather publish that because they know that the books will sell. On the other hand, if someone comes along and says, oh, that's wrong, here's my idea, and my idea I think is better because it's supported by so much evidence, they won't publish it. And the reason they won't publish it is they won't sell it because universities don't like controversial stuff.
Yeah, yeah.
And so it's dead in the water.
Yeah.
This is a problem.
Yeah.
And that's why textbooks, and the ones I'm most familiar with, are the biology textbooks, and they'll tell you in terms of water, which we're going to get to, they'll tell you that the water inside the cell is just like liquid water, but it's not. And the way you can tell that it's not, very simply, take a raw egg, you know, reasonable cell or pre-cell. Look at the egg white. Is that a liquid water? Raw egg white?
Yeah.
No, it's not. It's a gel. So, you know, there's an example. Another example is if you're brave enough, take a razor blade and run the razor blade along your forearm, and so you know what happens. Blood comes out. Water doesn't come out. But if the cells were filled with water, then you get the water emerging like from a breached water pipe. Yeah. It doesn't happen.
No.
So simple observations can tell you that the cell.
But we're good.
Okay.
Just keep going.
You know, that the cell is not an aqueous solution, not really this water. It's different. And we started from that point of view and went on from there. But I'm digressing.
Yeah. Amazing, amazing. But yeah, before we get into that, because obviously we're definitely going to, I just quickly want to touch on, so you mentioned Sir Andrew Huxley, and correct me if I'm wrong, his theory was the sliding filament theory, right?
Well, it's sliding filament rotating cross bridge theory.
And so that is currently the theory that we're learning in class.
Absolutely.
In biology classes. I had a cellular biology class last semester. We learned about the sliding filament theory. My AP bio class in high school, we learned about the sliding filament theory. And, you know, when you learn about these things in a classroom, right, you're hearing it from a teacher, and you think, okay, whatever comes out of the teacher's mouth is correct. It has to be correct, because I'm paying to come to college, and I'm in this esteemed education system, and whatever I'm learning must be correct, right? Yeah. There's no way there's other options. And also, when you learn, let's say, the sliding filament theory, you're not told that, like, maybe this is wrong. There are other theories, you know? And I think that's like where the problem lies in science education. Would you agree with that?
When you say that's where the problem, I'm not sure what that is.
In the sense of, like, you're being taught that what you're learning is like the only option and the only theory.
Yeah, absolutely. And the teachers who express those points of views, they're not at ease tearing it apart and suggesting it might be wrong, even if they might think so, because you know, it's not the thing to do, and they'll be criticized by their colleagues.
Exactly.
So the system system is built in a way to reinforce ideas whether they're true or not true.
Yeah. Have you ever taught like a intro bio course?
Well, not intro bio, but something equivalent to it, and the students loved it.
Yeah. And how do you go about teaching a course like that? Like, so do you, like, I guess, do you follow the standard textbook?
No, I never do. No, no. I present the textbook view, then I explain why it's wrong, and usually they see right immediately that the ideas are, are true, correct.
Yeah.
And then I tell them what I think might be the truth, and of course, there are some conjectures involved with that, and they might or might not accept it. But I've been very successful. In fact, so successful that the powers that be, the people in my department who don't like to be challenged, they eliminated the course.
Wow. Well, that's unfortunate. That is
no, well,
unfortunate.
I mean, it's a sign that something is going on, because there is no reason to eliminate the course.
That's true.
So they don't, in terms of water, there are lots of people, as you might imagine, in a scientific department that deal with water, because water is central for everything, much more central than they think. But water's everywhere. We're 2/3 water, and if you translate that into the fraction of molecules, like if you line up all the molecules in the body or in the cell, more than 99 out of 100 are water molecules, because they're really small. To fill that 2/3 volume, you need a lot of them. And so the current view is 99% of our molecules don't do much.
Yeah.
It's kind of,
yeah.
Amazing.
All right, so let's get into it. I do want to get into Gilbert Ling's theory, and we're going to talk about that. But I feel like a good starting place is Harold Hillman. So I have one of Harold Hillman's books here. You're familiar with Harold Hillman, and he essentially postulated that I know he talked about the endoplasmic reticulum and nuclear pores as being artifacts of electron microscopy. He also mentions the cell membrane, and so maybe let's start with the cell membrane. Does the cell membrane exist?
Well, you ask penetrating questions.
Yes.
According to Harold Hillman, it's an artifact of electron microscopy, and well, let me explain a little bit. So, if his argument. So, early on, the reason that cell membrane was conceived, there's no way of seeing it early on before the advent of electron microscopy. The inside of the cell has lots of potassium, and the outside has lots of sodium. And so people conjectured that there must be a barrier of some sort, and that's the cell membrane. And there was no evidence for that until the advent of electron microscopy. And then electron microscopy came into being, and sure enough, you could see if you took a cell and prepared it, all the preparatory steps necessary to do electron microscopy, which is like a dozen different steps. And those steps, I wouldn't say kill the cell, but they not only kill the cell, but they do all kinds of egregious insults to the cell. And then you look at a thin section is what you look at. So here's a table, and you look at a thin section like this, and you look down on it, and what you see are two tracks. They look like railroad tracks that run along the edge. And everybody was quick to hail this as evidence that those two tracks are the cell membrane. Now, Hillman says it's possible, but it's also something that's weird, because people who made the measurements have measured that the distance between one track and the other track is a consistent figure, 10 nanometers approximately, and lots of people are reporting 10 nanometers. He said, but that's impossible, because think of it, you take the cell, let's say the cell is a sphere, and the section that you choose, let's say, runs right through the equator, okay, and then you look down, and you should see the proper dimension, that is from one to the other should be, let's say, 10 nanometers. But you don't know if you're sectioning right on the equator of the, let's say, sphere.
Yeah.
So if you're sectioning at a different region of the sphere, let's say right near the pole, to exaggerate, then you're looking obliquely. You should be looking obliquely at the membrane, and the spacing should be larger between those two tracks.
Yeah.
But it's not.
But it's not.
It's not. So Hillman suggests that there are so many steps in the preparation for electron microscopy, and you should get this distribution of distances between those two tracks, but you don't.
Yeah.
Now, so he says it's a good chance that the membrane, the main evidence, maybe the only objective evidence, is wrong. Besides that, modern biology suggests that this membrane should be interrupted by pumps and channels that are supposed to exist in the membrane, but you don't see them. All you see are these two lines.
Yeah.
So no, my own view on this is that his work needs to be repeated because you know, there's a possible bias that exists. Everybody knows that the distance should be 10 nanometers, and so if you find a distribution of distances, you publish 10 nanometers, not 15 nanometers or 20 nanometers.
Yeah.
You know, and it's called what is it, experimental bias, or some, I forget the term that's used, and you can understand that would occur. So my view is I don't know the answer. Harold Hillman, bless his heart, you know, he brings up matters that are really pertinent and really important, and they need to be checked.
Yeah, and he firmly believed in what he was in his theories and his conclusions. So, it definitely makes you question, like, do we really know the answer to it? Awesome. Okay, so I think that's a great introduction for Harold Hillman. Let's get into Gilbert Ling quickly. If you could, maybe how did you discover Gilbert Ling, and what was your first?
Well, I actually discovered him. He was in Philadelphia for some time, and I was a student in Philadelphia. And I remember sitting at a dinner somewhere, and the guy next to me said, "Hey, that guy 3 tables over, that's Gilbert Ling. He has this weird idea of water in the cell." And that's all I heard for many years. And then one day, I was invited to a conference where I met Gilbert Ling. And the conference was, it was in the memory of a guy, a scientist named Ernst, who passed. And it's a memorial symposium for him. And Ernst was interested in 2 things, muscle and water. And I was asked to represent muscle because he, just like myself, he didn't believe in the sliding filament cross-bridge theory. And so I went there, I gave my talk, and I guess it went reasonably well. But I met Gilbert Ling, and his presentation just blew me away, absolutely blew me away. He was talking about water inside the cell, and he said the water inside the cell is not like liquid water. He said that the water molecules were lined up like soldiers at attention, right? And how could that be? Well, if you think of a water molecule as a dipole, like a bean with plus at one end and minus at the other end, you can imagine how these beans would stack. And physical chemists generally believe in this stacking by 1 or 2 molecular layers, and he was talking about hundreds of molecular layers. Later we found up to millions, something not exactly as Gilbert was suggesting, but generally a similar kind of idea. And he presented lots of evidence for it. And you know, and then other people had been invited to the conference, and they provided additional evidence. So, you know, I'd been in the field of muscle contraction for 20 years, or even a little longer, and I said to myself, muscle is really important, but water is even more important, and this is really fascinating. So I came back and I gave one of those books, I forget which one, you've got a couple sitting there.
Yeah, exactly.
Yeah, one of the books to some of my students who I thought were particularly astute, and I said, hey, could you give me some feedback? I'd like to, what do you think about this stuff? And their feedback was the same. This is a really interesting idea, and if it's true, that means all of biological thinking is not true. It's that profound because, you know, we think of, if you think of the water inside the cell as ordinary liquid water, things can diffuse around and such. And in this case, they can't. It's a bit like a crystal, like a liquid crystal, or like ice, not exactly ice. Not much can happen in there. And you know, if you have ice, reactions can't take place because things can't move around and such. So they said that it's really basic and profound in thinking about life and thinking about the cell. And also, the evidence that he presents in there, though complicated, looks really convincing.
Yeah.
So I couldn't help, but I was mesmerized. But I understand that I'm easily mesmerized. so I wanted to check myself, and they confirmed.
Yeah.
So what do? Well, you change fields and you start studying water. We had no money to do anything with water because in order to do experiments, you need money to hire people, and pay their salaries, and get supplies and equipment and such. So I decided what I'm going to do is write a book.
Yes, and here it is.
This is the book that I wrote, Cells, Gels, and the Engines of Life. Cool title, nice cover.
Awesome cover, I love it.
Yeah, but it was received with mixed enthusiasm. You know, some people said this is just Gilbert Ling, and everybody knows that Gilbert Ling is a crackpot, and therefore pay no attention to it. At the other end of the spectrum, there was a well-known cell biologist from Harvard,
Don Ingber.
Yeah, right
there.
Oh, so you got,
yep,
the review.
I have his review, and it was amazing.
Yeah,
he loved it.
Yeah, you're conscientious. He said it's a 304-page preface to the future of cell biology.
Yeah.
I like that one better.
I like that too. I wanna dive a little deeper into Gilbert Ling's theory. So he came up with the Association-Induction Hypothesis as opposed to the membrane pump theory, which the membrane pump theory, for people who don't know, well, one, it's taught in every single biology class around the world, as far as I understand, all the biology classes I've ever taken. And essentially, it postulates that we have this fluid mosaic membrane where there's proteins, that these proteins can move around in the membrane, and these proteins are pumping in and out solutes.
Yeah.
Primarily, we can think of like sodium and potassium.
Yeah.
And Gilbert Ling essentially says this theory is wrong, and he says I have a different theory and hypothesis, and the Association-Induction Hypothesis. If you could kind of run us through that hypothesis as simply as you see it.
Well, I mean, the essence of the hypothesis is what I described earlier, that the molecules are lined up, and I think that hypothesis is it's not exactly independent, but it's a different subject. It's different from the subject that you were mentioning about the pumps and channels.
Okay.
Now, he dealt with pumps and channels. Pumps. He dealt. The pumps are logical because there has to be a way, one would think, of explaining why the cell has more potassium and the outside of the cell has more sodium. Mm-hmm. You know, if you were naive and thinking about it, you'd say, well, there must be a pump in the membrane. So Gilbert found that there's not enough energy, and this was a time when the only pump was a sodium pump, so-called sodium pump. Mm-hmm. Now there are, according to one of my students, more than 1,000 pumps for all kinds of different things. And so if there's not enough energy by far for the cell to pump those ions, you multiply that by 1,000, and you know it reaches the point of absurdity. Mm-hmm. But Gilbert did more experiments. He would cut the cell, mm-hmm. you know, and if the pumps are necessary for the cell to live, you're basically short-circuiting the pump, the action of the pump. But the cell does fine. It doesn't generate, other studies have shown, it doesn't regenerate the membrane, but the cell keeps living, you know?
Yeah.
So there's something that's absurdly wrong with the hypothesis.
Yeah. And for people who want to like really dive deep into this, Gilbert Ling's website is full of amazing information. I printed out one paper where he basically has, it's titled 3 Sets of Independent Disproofs Against the Membrane Pump Theory. One of them that you said, you strip the membrane away and the cell acts normally as it would. The one I'm really interested in is his first disproof, in that, as you mentioned, there's not enough energy to operate this cell. And one of the papers that he references is the Podolsky and Morales paper, where essentially they're looking at the original paper that says ATP, adenosine triphosphate, has this high energy phosphate bond, and that energy is what is powering all these processes in the cell. And then Podolsky and Morales comes along, and they're saying that that paper needs to be revised. There is actually no high energy phosphate bond in ATP. And so, as far as I understand, maybe correct me if I'm wrong, no one has said this paper is wrong. Like no one has disputed.
Yeah, I know those two guys,
Okay.
Morales and Podolsky, because they're in the muscle contraction field. And I almost did a postdoctoral fellowship with Podolsky. Instead, I went to Seattle to take a faculty position. They're very competent people. So this is a problem, and it's one of those problems that needs to be addressed. Again, some people have to repeat these studies and see what they get. It was, I think, on Gilbert Ling's original website. I'm not sure if it's on this one, because some people have revised it. He talks about a paper that appeared 1 or 2 years after the famous Lippincott paper that was 1941, I believe, that said ATP has a high energy phosphate bond, and that's where we get our energy. You know, if we're playing basketball, we need energy, you know, and that's where it comes from.
Yeah.
Well, so there was another group referred to by Ling, and I haven't seen the paper myself.
Okay.
And they said these guys, Lippincott and prominent scientists, they made a simple arithmetic error. There is no such thing as a high energy phosphate. And then comes Morales and Podolsky, and they don't directly say this is a challenge.
Yeah, they don't.
Yeah.
They actually say this does not threaten the energy donor role of ATP, which I guess I kind of understand. Like essentially, there still is some sort of energy donor, but but they do say it's significantly smaller than what we thought.
Yeah,
right?
Well, knowing these two guys, they would be timid and that wouldn't want to overstate the case. And you can imagine, paper goes under review, and the critics or the reviewers say, well, you can't go to such lengths. So this ATP story has been around for quite a few years, and everybody believes it. Therefore, don't be so brash in your conclusions. And then they have to change it. So I'm not sure what happened exactly, but this is a critical paper.
Yeah.
And there are a few others besides those two.
Yeah.
So, you have to ask, well, if the energy doesn't come from ATP, then where does it come from? And as we talk about our experimental results and such, there is another option, and that option is electrical energy. Yeah,
and we'll totally get into that very shortly. Like one other last point that I want to add here is that Ling says in this writing here that the energy, the minimum energy required in the cell is 600 to 1,200 times greater than the energy actually available in the cell. So
you mean for pumping?
For pumping, yeah. And this is just for the sodium pump.
Yeah, right.
That's one pump.
Yeah.
And like you said, there's
more than 1,000.
More than 1,000, yeah. So Yeah, it's crazy.
It's one of those things, you know, it gets embedded in the literature.
Yeah.
Everybody reiterates it, all students learn it, but it's not right.
Yeah, yeah. Awesome. All right, is it fine that the sun's...
I love it.
Okay, awesome, perfect.
Yeah.
Cool, getting more infrared light. All right, so I think that sets us up very well for getting into the work of your lab over at the University of Washington. So I guess let's just start. Well, actually, sorry. Let me step back a little bit because I do have one other thing about ATP. So I heard you in a podcast saying that potentially ATP is not this energy source of this cell.
Possibly.
Yeah, possibly. And if it's not, I heard you talk about. I think Gilbert Ling says that ATP is a cardinal absorbent. What does that mean? Cardinal absorbent.
It means that structured water builds.
Okay.
That's a kind of nucleating site for the buildup of structured water.
Got it. So potentially, and we'll get into it. Potentially, that it would be the phosphate group, the negative phosphate group.
Yeah.
So easy, let's say, could potentially build from that phosphate group, and then that is actually where this energy.
That's
may be
what he
coming
was
from.
suggesting.
Okay.
Yeah.
Interesting. Obviously, he wasn't saying easy, but he had this other sort of idea. But okay, cool. Good to know. All right, so let's get into this book, The Fourth Phase of Water, which is an awesome read.
Thank you.
Yeah. So I think we should start with Gilbert Ling's idea, and you mentioned it, stacked dipole. And why don't you tell us how you found that potentially this is not the case?
Well, dipoles are neutral. You know, you got a plus at one end and minus at the other end. So you can stack dipoles from here to the moon, and you'll never get net electrical charge. It's going to be neutral, whatever. And so Gilbert Ling's idea implicitly proposes that his so-called structured water, which we give a somewhat different name, is neutral. But we found it's not.
Yeah.
So we found. Well, maybe I should.
Yeah, we can
go to what we found.
Yeah,
and then,
let's get into it. Yeah,
so,
perfect.
So Gilbert Ling's. We first followed along Gilbert Ling's ideas because we wanted to essentially confirm that he was correct, because it looked as though he was correct, and we thought we'd use different methodology to check it out. So. If the water molecules are organized the way he was suggesting, it's sort of like a crystal, so-called liquid crystal. And for a liquid crystal to be pure, if you start with water that's let's say not pure, you know, there may be some contaminants or ions or whatever. In order to form a pure crystal, as he was suggesting, you got to get rid of those contaminants to exclude them. That's where exclusion zone came from. So we took a chamber. This is not in biology. It's a laboratory demonstration. We took a chamber. We put water, and we put little particles that we thought might be excluded. Little spheres called microspheres, and they're commonly used. There's nothing exotic. And in that chamber, with microspheres suspended all over, we plunked. The first experiment was a gel, a hydrogel, a gel made of water. You know, modest-sized gel. We plunked it in the water, and we looked to see what happened at the boundaries of the gel. And the first experiment worked. And I mean, we found that these microspheres were eliminated from all regions surrounding the gel, and not just a few molecular layers, but something like well, I'd say how many molecular, it would be hundreds of thousands of molecular layers, you know, a few tenths of a millimeter, big enough you could see it with your naked eye. And it took 3, 4, 5 minutes for these. So this is the surface. The surface, here's the gel, and here's the surface of the gel, and here's the water with the microspheres. And what we noticed, we saw that this exclusion zone, the zone where that's how it got the name, where the microspheres were excluded, would grow and reach a few hundred micrometers, and then stop growing. And so that was confirmation for us that there's some kind of ordering that goes on. But we didn't know. We assumed that, yeah, Gilbert Ling is right, because the ordering has maybe something to do with the water molecules stacking. But then we did another experiment, and I'm not sure what motivated us to do that experiment. Maybe previous work that I had done, sticking micro electrodes into cells and measuring the electrical potential. And in order to do that, this was another invention by the same Gilbert Ling. He invented a so-called microelectrode, which is nothing more than a glass coming tapered to a point with an opening, fill it with potassium chloride, and stick it in the cell. And you could measure minus 50, minus 60, minus 70 millivolts, and you could pull it out and the cell didn't die. It was really an invention, a powerful invention that won other people Nobel Prizes who used that technique, somewhat modified, but Gilbert never got his Nobel Prize, unfortunately. I think maybe because he was controversial, and controversial people don't get Nobel Prizes usually.
Yeah. And this is a side note, but his theories went on to basically make the MRI, right? His theories were used
correct.
for the MRI?
That's
okay.
correct.
Yeah.
Yeah.
Very,
it was
very
someone who worked in his laboratory, a pathologist, who was an expert at, or wanted to learn nuclear magnetic resonance, and he went on to get the patent on MRI. And he was passed over in terms of Nobel Prize because he's a controversial guy. But yeah, so a lot of things came from Gilbert Ling. And well, at any rate, I knew about these electrodes, these microelectrodes. We had used them to stick into cells earlier in my career. And I felt I knew how to do it. I thought, well, gee, why not stick it into the exclusion zone, or easy to remember.
Yeah.
It doesn't work in Europe because EZ, but easy water, easy to remember. Yeah,
I love it.
Yeah, well, it was fun. It was not an original suggestion. It was a colleague from Australia. He said, well, why do you have to go through all the description and background? Why don't you just give it a name?
Yeah.
Yeah, and he said, call it exclusion zone, or EZ.
There we go.
Yeah, that was. Okay, so we stuck the electrode in, and by golly, to our great surprise, we found that it was negatively charged. We expected zero because, you're starting with water next to one of these surfaces, a gel surface or polymer surface, hydrophilic surfaces. You start with pure water, and pure water is neutral.
Yeah.
So you start with pure water, and you should end up with pure water, but it was negatively charged. And then we thought, well, something's wrong here, because if you start with something that's neutral, how do you get something that's negatively charged?
Yeah.
Doesn't make sense. The only possibility we could think of is there must be another region that's positively charged, because you add negative and then positive, and you get neutrality. Mm-hmm. So we found, indeed, that you start with a gel or any of various other surfaces, and next to the gel you get this EZ or exclusion zone that has negative charge, and beyond it you have positive charge, spread out in the liquid water. So now Gilbert Ling's idea is different from that. He said it's water molecules, but it can't be water molecules because they're neutral, stacked water molecules. And that's when we started thinking about battery-like effect, because if you've got separated negative and positive, you got a battery. Yeah. Right? And batteries give energy, So we're thinking, well, is it possible really that water, you start with water and you wind up with a battery, basically a water battery is what you got. And parenthetically, if this is the water that's inside the cell, then you got a battery essentially. Yeah. And so batteries can produce electrical energy, and if the ATP story is wrong, or even if it's right, still there is the possibility that this battery can contribute energy. Not necessarily all the energy, but some of the energy. Or if the ATP story is wrong, then possibly all of the energy.
Yeah.
So anyway, that's how we got started, and I think it's probably best if I respond to your questions instead of going on and on.
Yeah, let's. Since you mentioned the battery, I had a couple of questions on this. So you guys actually found that there was a potential difference between the positive and the negative,
yes.
and you had this current running. You had like a current basically, right?
Well,
yeah. Correct me if I'm making
no.
any mistakes.
You don't have current yet. You could have current, but well, to start with, you've got what seems like a static situation with negative and positive, just like a battery.
Yeah.
You know, you got positive and negative, and this is sustained for some time. But, you know, you can't create a battery. All batteries need charging. You can't create energy, potential energy from nothing. You have to have another kind of energy that is transformed into this kind of energy, and so the question is, well, where does this energy come from? And we couldn't figure it out. I spent 2 years scratching my head, lack of hair, figuring out. I couldn't figure out the answer.
Yeah.
And then the answer came from someone just like yourself, whose curiosity, I guess, is maybe comparable to yours, or he was bored. I'm not sure which. And so he's doing an experiment like the one I described to you in the chamber with the microspheres. I can't remember what he was doing, but I don't know, as I said, either he was bored or curious. He saw right next to him was a lamp, an incandescent lamp, a gooseneck lamp. He took the gooseneck lamp and he shined it on the chamber. And he called me in to show me the result. And the region that was illuminated, the EZ, grew by 3 times approximately from illumination. So he called me in, and I said, "Wow, this." I'm thinking he's got the answer, and it's light that's doing it. And I quickly thought, what about photosynthesis? This is similar because photosynthesis, the energy comes from light, and the first step in the 20 or so steps of photosynthesis is a separation of negative and positive charge. Mediated by light. So I thought, well, we got something that's, similar to photosynthesis. So there's good precedent for it. And then we wanted to find out which wavelengths are important in doing this, because incandescent lamp contains a series of wavelengths ranging from the UV through the visible spectrum, and at the long wavelengths, infrared. And we tried UV light, nothing. Visible light, some effect, but pretty minor. And we got to the infrared, to the longer wavelengths. It was like bonanza, gangbusters, just a very tiny amount of light could grow the exclusion zone enormously. So we found the answer. Now, a lot of people don't know infrared light. Well, that's something that you see in the toaster or the oven. It glows.
There's red glowing.
Red glowing or orange glowing, and generating heat. And you say, oh, that's infrared light that's coming from there.
Yeah.
But infrared light is all over. It's being generated. So if we were
being bathed in infrared light right now.
Well, from the sun, roughly, I can't remember, it's 49% or 51%.
Yeah, it's around 50%. Around 50%.
About 50% is in the infrared region, not in the visible region. So, but also, it's not just the sun. It comes from just about everything around. It absorbs light and then regenerates. So if we were inside, and if you were to turn off all the lights, so you can't see anything, and your cell phone camera can't see anything, if you take out an infrared camera, which is just like a regular camera, but the sensor is sensitive to the long infrared wavelengths, not the visible wavelengths, which are shorter. Complete darkness, infrared camera will give you a beautiful image of everything,
yeah.
which means that everything is generating infrared light.
Yeah.
And that's why military uses it to look at the enemy tanks at night, you know, to see what's, and rifles and whatever, because everything is generating infrared.
Yeah.
So,
yeah, I was trying to, I guess I didn't bookmark it, but in the book you have a diagram, or a graph I should say, of once you shine the infrared light, how dramatically the EZ increases, and it's really dramatic. I know you said like three to 5 times.
Well, 3 times. We could get it up to 10 times with very weak light.
Yeah, exactly. And then, so on that same graph, there's, I guess, like a predictive graph of if you were to be using a much more stronger infrared light.
Yeah,
right?
yeah.
And that shoots up to like 50 or somewhere up, or pretty high.
Absolutely.
Yeah.
Yeah, absolutely.
It's amazing. Yeah. So, so awesome. I think that does a good job with the battery.
So I mean the finding of a battery-like operation, what charges the battery, and just like the first step of photosynthesis, light.
Yeah, light. Actually, good thing that you brought back up photosynthesis. I know you're familiar with Arturo Herrera, the researcher down in Mexico who studies what he calls human photosynthesis. Yep. Obviously, different idea than what you're proposing, but it's really interesting. So I got our library to purchase his book because I was I guess you can say skeptical, and the library purchased the book. I read through some of the book, and I guess just to preface, you know, I think there might be a language barrier. I was reading through the book, and at least it's not perfectly written in English. So that's one thing, and two, I guess there's not, as far as I understand it, he doesn't have like too much solid evidence for it, let's say, at least for melanin splitting water molecules via light. I don't know if you've seen other evidence for that.
He sent some of his melanin to our lab,
okay,
to test, and we were thinking maybe there's an exclusion zone next to the melanin. We couldn't find it.
Okay.
So I'm not sure. You know, his approach is a little bit different from the usual scientific approach. He's a very enthusiastic guy, and he might really have something there. I'm not sure, but we couldn't find evidence for it.
Yeah.
But he seems to be exploiting it for to produce energy, and he says he's successful. In fact, he's so successful that in his office or laboratory or whatever it's called, the Center for Human Photosynthesis, he now has a gate around it. It's locked.
Really?
So you know, you can't get in to see what exactly he's doing unless he wants to show it to you.
Okay, interesting.
He's a wonderful guy. He invited me to his daughter's wedding, and
nice.
I love the guy. I'm just not sure.
Yeah, okay, makes sense. And I mean, look, I'm not saying what he's saying is correct or incorrect, but I mean, it would make sense that through evolutionary history, we would have had this idea of photosynthesis would have been conserved into animals, at least using sunlight for some energetic purpose. And what you guys have obviously, as we've explained.
Yeah, I agree 100% with you. And so my current thinking is that what we've discovered is a generality, and photosynthesis is nature's expression of that generality.
Yeah.
Probably the most efficient, the most effective, because nature does things in the right way, in an effective way.
Yeah.
So photosynthesis then is a kind of subset of a more general phenomenon that I think we've identified.
Yeah. Amazing. Awesome. Okay, and we've hinted at this, but so essentially this EZ water that you guys found, laboratories across the world have replicated these studies, and they've found the exact same thing, correct?
Yeah.
And correct me if I'm wrong, but actually before you guys even found this, there was a study like way back when that found this exclusion zone water, correct?
I'm not sure.
Okay.
Maybe you need to refresh me on if you have details.
I don't. I just remember. I don't know. I remember hearing that. I guess I'm pretty sure you said it in maybe in one podcast that like to your surprise that there was a group of researchers who did something similar and found this exclusion zone next to a hydrophilic surface.
Yeah. It's called Polywater.
Polywater. Okay.
Yeah, the Russians.
Okay.
They found something. They didn't find exactly, or they didn't present it exactly in the way we did, but they found a different kind of water, this water could be found in thin capillary tubes, and the boiling temperature was higher, the freezing temperature was lower, and it had peculiar properties, and it behaved more like a polymer of water than isolated water molecules, and they called it polywater. And then came the so-called polywater debacle, because it was done by Russians, and at the time, the Russian stuff was not translated into English, so almost nobody knew about it.
Gotcha.
Okay. And then the translations began during the Cold War, and people say, "Wow, this is amazing." Either it's true and amazing, or it's not true. And many of the people thought, well, it's not true. It can't be true because we know everything there is to know about water, and therefore, this is can't be possible, can't be true. So various people decided to study it, and a few people came to the conclusion that it's an artifact. It's not real. And there was one group in particular, I think from Australia, and this group in Australia was saying that if I can remember correctly, I don't talk about this stuff very often because it's old hat, and we don't know whether it's actually the same as easy water or not, but it might well be. And they thought if they put salt in their water, they found they could get pretty much the same result as what the Russians got apparently for pure water. So they said they must have been sweating and doing experiments in the summer, and the sweat got into the water and produced this. It was kind of like a scientific joke, polywater nonsense.
Yeah.
And it was welcomed by the research community, because the research community in general is rather conservative. They like to think that what they know is the truth. And if someone finds something in conflict, that it's probably wrong, can't be right. They like to defend their turf. So it's not clear whether polywater is real, and if it's real, whether it has similar or the same properties as what we've measured, but it's quite possible.
Yeah. Okay, cool. I want to talk about the structure of bulk water versus easy water. In the book, you detail this really, really well. But essentially, at least from my reading and my understanding, that easy water is more of in the shape of ice. You can say, at least you guys started with the premise. All right, let's do this hexagonal crystalline structure of ice, and then let's go from there. And then you've obviously it's not exactly like ice. So if you could walk us through that thinking and what is the structure of easy?
Well, the structure that we deduced doesn't necessarily mean it's correct, but we think it's correct.
Yeah.
Like all the science, you never know.
Yeah, exactly.
But it fits a lot. So one of the key features is X-ray diffraction. And X-ray diffraction can give you an indication of what the atomic structure is, or molecular structure. And a group from Harvard University actually did X-ray diffraction of water that they thought was similar to easy water, and I think so too. And they found out it was hexagonal. So you start with that, and then there was another group who also dealt with the idea of some kind of structured water, hexagonal water, and this was they were working with the Russian findings of polywater, and they also thought that it's got to be a hexagonal structure, sheet-like structure. So we took those two observations and a few more, and we basically came to the understanding that this thing builds in hexagonal sheets. So each sheet has a hexagonal motif to it, and as you know, hexagons appear everywhere in nature. So it's not a surprise. And ask me, because I'll forget, what it is, a special feature of hexagonal water that's critical for an understanding of how the charges are separated.
Yeah, so essentially, if I'm correct, like the hydrogen, which you can think of just as like a proton, gets basically taken out of the hexagon, right, and gets pushed into the bulk water?
Right.
Would that be?
Well, there's more to it,
Okay.
because, you know, let's say that's the case, and I do think that's the case. So you've got negative next to positive. Well, you know, it's sort of like man and woman. They're attracted to one another. And and it's really virtually impossible to separate positive and negative charges, right? They immediately want to recombine.
Exactly, yeah.
Right? They want to merge together. And so if you have a battery, or if you have charges that are separated for some length of time, there's got to be a mechanism to keep them separated. Well, if you think about the EZ having a hexagonal motif, in order for those positive charges out here to enter, they really want desperately to enter this EZ, they have to pass through those holes in the hexagon. Now, the protons that are out here immediately latch onto water molecules, and they become a so-called hydronium ion. And this is not my conjecture or something. This is in all chemistry books. So you have these hexagonal openings that are very small, and these hydronium ions that have an opposite charge and want desperately to get into this matrix, but they can't enter those holes because they're too small. So they're kept out.
And that's because the hexagons basically like overlap to a degree, right? So the holes are actually getting smaller and smaller when they're overlapping?
Absolutely.
Okay.
Yeah. So that reduces the size. So if you have just one sheet with a hexagon, it's pretty small. But the way EZ is arranged, each sheet is slightly displaced from the next one.
Exactly.
So those holes are smaller.
Yeah. And so if you were to leave like this, let's say you take a hydrophilic surface, put water in there, this EZ grows. If you were to leave that and have no infrared light being absorbed, then would those protons then diffuse back into, or maybe diffuse is the wrong word, but go back into the EZ, and then the EZ would get smaller from there, right?
Yeah, I think that's what happens. I mean, inevitably all systems run down. You know, you try to. But we're talking about running down over a period of hours, or maybe sometimes even days.
With no infrared light, are you saying?
Well, with minimal infrared light. Even with some infrared light, there's a tendency over a long period of time to run down.
Okay.
Now,
why do you think that is?
Just because no system is perfect.
Okay.
You could have one sheet of EZ, one layer of EZ, and maybe it breaks apart or something, and then lets the positive charge come in. Something like this. You know, I mean, any system of energy cannot be preserved indefinitely. Something just doesn't work the way the theory suggests, and eventually it peters out. Yeah,
makes sense. I think what's really interesting is that, so when I first started getting into your work, and I heard infrared light builds EZ, I guess I thought infrared light was, for lack of a better word, more important in the sense of, so infrared light basically just, if you have just water molecules and they're hydrogen bonded to each other, infrared light splits those hydrogen bonds, correct? And then that is when EZ can start to form.
Yeah, right? I mean, that's a conjecture, but you know, it's the most reasonable conjecture that we could come up with.
Yeah, okay. I noticed, at least, so I haven't gotten through the entirety of this book, but I got through most of it. And you never talk about hydrogen bonds. You reference it, but don't say it by that name. Is there any reason for that, or no?
Well, I'm not sure it's correct.
Really?
Yeah.
Okay. Please I would love to hear.
Well, no, I don't necessarily have, any indication of that. Just it's a conjecture, and I would like to see evidence for that conjecture.
Yeah.
Maybe it's absolutely right, but I'm not sure. Like everything else, without direct, clear evidence for something, I can't be sure. And because now we know about the existence of EZ water, you'd have to go back and look at whatever evidence exists for the hydrogen bond and see if that really is a reflection of EZ water.
Gotcha.
And I haven't done that because there's been no motivation particularly to look at hydrogen bonds. It's one of those things, you read about it all the time. It's in every textbook, and you need to go back and ask the question whether it's right.
Wow, so interesting. I mean, I would never even think about questioning hydrogen bonds. It's so ingrained.
Well, then you would never think of questioning the structure of the atom, would you?
I would not, but I know you have.
Yeah, I've got a book that's ready to be illustrated. My son is the artist, and he takes his time. And so he illustrated the fourth phase of water. A lot of people commented. He's a fantastic artist. And the book that is just coming out, we just sent it to the printer. It's called Charged. The subtitle is The Unexpectedly Profound Role of Electricity in the Workings of Nature, or something like that. And we're pretty excited about that.
Do we have a date of when that's coming out?
Well, yeah. I mean, since we sent it to the printer said it takes between 2 and 3 months for them to go over details that are maybe not so clear, to print it and to ship it from South Korea, where it's being printed, to our distribution center. So it means kind of like the end of May or beginning of June.
Okay. All right. So coming up, and I heard. So that's one book. I heard you have either 1 or 2 other books coming out as well?
Yeah, I have. Next book, I'm waiting for my son to begin illustrating. He's actually illustrated the structure of the atom. Okay. And I think the current idea of the structure is incorrect. Yeah. And I can tell you a few reasons why if you ask me.
Please, yeah.
Okay, and then I'll get to that in a moment. Another one that is in progress, almost done, is volcanoes and earthquakes. Where does the energy come from?
I don't know.
No, well, a lot of people don't know. I think I have an idea. But let me get back to the structure of the atom.
Yes,
so that's been around for, we think it's like the solar system. Even with the addition of quantum mechanics, if you think that's correct or not correct, I have something to say about that too. I don't want to go too far astray, but think about, I mean, even with the addition of quantum mechanics, still you've got a nucleus that has positive charge, and electrons that are in orbitals around the nucleus with negative charge. So just think about that for a moment.
Yeah, the electron should collapse in on the nucleus.
Exactly.
Yeah,
but
it doesn't.
It doesn't. Well, according to the model, it doesn't. Yeah. And when Niels Bohr was asked, what about the hydrogen atom, where you've got positive nucleus and one negative electron, how come they don't merge together? He said he's famous for having said they just don't. And that for me that's not
sufficient. Yeah.
As important a scientist as he was, and then there is the other argument that has been addressed. I'll get to that in a second. So the nucleus, think about a nucleus, has got a lot of protons and neutrons, and the protons are positively charged and they're packed closely together. When you pack positive charges close together, they want to explode.
Yeah.
Right?
Yeah.
And so the physicists, they did realize that that's a problem, and they had to do something about it. So they invented something called the strong force.
The strong force. I've heard about this. Yeah.
Yeah, and there's no. I've never heard of any evidence for a strong force, except that we know the model of the atom is correct, and therefore there has to be something like a strong force.
Yeah.
For me, this is like a Band-Aid to cover a gaping wound.
Yeah, I agree.
Yeah. So you got got two fundamental problems. The nucleus is going to explode, and the electrons are going to collapse. So it's unstable.
Yeah. What is there evidence that the electrons don't collapse? Besides, I don't know. Do they have?
No, I've heard nothing. No evidence. It's just that that's the way it is. The strong force is a very important fundamental force of nature because A, we know the model is correct, and B, in order for it to be correct, the nucleus can't explode, right? Or the electrons can't collapse. If you said, what's the consequence if the electrons collapse? Well, if that's the case, then the atom collapses into a point, to nothing. Which means that this table made of aluminum or whatever collapses into one vanishingly small point. So
and at that point we wouldn't even be able to see the table, but we see the table.
Anything, so. Right? Yeah. So I took it upon myself to think alternatively, and it's too much to go into, but I can say that I spent several chapters. If you start with the model that I'm suggesting, which is actually not so complicated, very simple, you can explain physical phenomena in a very straightforward way. And one more thing. Maybe you're not aware, maybe you are aware because you're aware of so many things in science. I'm really impressed.
I try.
Well, I'm really impressed.
Thank you.
That Niels Bohr was a physicist, and early 1900s when he came up with this model, it was the era of physics, right? There's Einstein and Max Planck and Niels Bohr and so many of those people who all won Nobel Prizes and et cetera, et cetera. And the previous century was the era of chemistry, but now it was the 20th century era of physics, and so the physicists were dominant. And this model was a model of the physicists. The chemists hated it. One of the famous chemists, G.N. Lewis, said, this doesn't explain even the simplest of chemical reactions. Can't be right. And he came up with his own model. And his own model is so close to what I came up with. I didn't know about his model until after I drafted the book. So felt pretty good to know that G.N. Lewis, famous for Lewis bases, Lewis acids and such, and considered one of the 2 or 3 dominant chemists of the era. He came up with a model that's not so different from...
Interesting. Would you be able to, like, I don't know if this would be possible, would you be able to, like, draw what the model of the atom kind of looks like?
I can tell you. Most of the atoms, I think, are cubic.
Okay.
And so if you want to put cubic atoms together, they fit very nicely. You want to put spheres together, they don't fit so nicely together, and they leave gaps. And so the question is, well, what's in the gaps? And that's not addressed.
Well, that's actually really interesting you bring that up. Like I remember when I was in like middle school chemistry, I found like a fun fact that like most of matter is nothing because of those gaps, right? Most of the matter in the world is nothing. It's just space. So, that's interesting. It doesn't make too much sense.
Well, it doesn't make too much sense to me, and doesn't make too much sense to you. I'm glad you concur. But so it'll be probably a year before that book comes out, because it just takes time. And my son on the next book, the charge book that's coming out, I offered to put his name on the cover, illustrations by Ethan Pollack. And he graciously accepted, but that meant it put the pressure on him to make sure that each and every diagram was absolutely optimized. And I think it is. The figures are absolutely amazing, and there are like 230 color illustrations in the book.
Wow.
They tend to explain and illustrate, and some of them are kind of fun cartoons. That you take a little laugh as you go through the book. And nothing needs to be perfectly serious. So it's going to take time before that that book actually emerges.
Got it. Awesome. Like, because I'm really curious about the whole cubic structure of the atom. So would are the electrons still spinning around a nucleus, or is there a nucleus? Are there electrons?
No nucleus, and there are no neutrons, and there are just protons and electrons. And in the book, I explain why. I explain why the evidence for the existence of those is actually equivocal.
Amazing.
And you can imagine stability is, for me, is the most important criterion. And you can arrange positive and negative charges in such a way that they balance one another, arrange them in space. And that's what I'm suggesting. And there is evidence for that, not necessarily evidence that occurs in the atom, but something called colloidal crystals, which you may or may not have heard about. Colloids, like milk is a colloid, suspended in liquid water, for example, and they are able to suspend themselves. Each colloidal particle has a negative charge, and if you have positive charges around them, they can balance so that the atom is stable, or whatever you have, the colloid crystal is stable. It's very stable. It says colloid crystal. It's actually very regular. And so my atomic model is based on that principle because it's stable. Negative and positive charges distributed in a certain way. And you can have small ones and big ones with more of those particles or fewer, and they balance one another.
Got it. Okay.
So that's the central theme.
Awesome. I love it. I'm super excited for the coming books, and I'll have to keep an eye out for them. Let's get back to EZ for a little bit. First off, there was this one study that you guys did that's talked about in the fourth phase of water, where you saw an EZ exclusion zone projection up to 1 meter. If I'm doing those calculations correct, that's like 39 inches. Is that,
well, approximately 39 inches.
Okay,
yeah.
That's like amazing. Because I mean, if we think about this and we tie this back into just cell biology, I mean, obviously we can't even see cells. Cells are extremely small, and so the majority of the water that is in a cell must be this exclusion zone water. If the exclusion zone is building a cell. But 1 meter that's amazing. That's crazy.
Well, yeah, the way we did this experiment, I say we, but it was an undergraduate student like yourself. And students are pretty creative. You get older, you get less creative, I think, at least for most people. So they were playing around with exclusion zones, and they thought, well, what happens if we have a long tube, like 1 meter long? And they took a tube, I'm not sure where they got it, it was like this long.
Yeah.
And they put a gel at one end and filled the whole thing with water. And they found that they got the usual kind of exclusion zone, maybe half a millimeter or something like that. And then what happened is that it began to diminish in diameter down to a so-called dendrite, which is like a long pole. And the long pole kept growing and growing, and it grew to the end of the chamber. And sometimes it would grow back again, and sometimes it would bifurcate, and you get a branch.
Wow.
And we took that as additional evidence for the crystalline nature, liquid crystalline nature, because there were some challenges to our interpretation. I can't say that I understood the basis for those challenges, but there were challenges. But it's hard to refute if you have something that grows like that, grows to 1 meter long. I mean, it's hard to come up with another interpretation other than it's a crystal or liquid crystals, which are known to be able to grow. So I could assume that the growth is practically infinite if it grows. So how many layers we're talking about? Let's see. So 1 meter, 1 micrometer. If the EZ is ordinarily, if each layer, let's say, is 1 micrometer, it's much smaller than that. If it's 1 micrometer, then you got 1 million layers. right?
Yeah.
And it's much less than 1 micrometer.
Yeah.
You know, so we're talking millions of layers.
Yeah, unbelievable. Why did it grow in that shape? Was there something different about the surface that was here, or is that just randomness, maybe? Or maybe we don't know.
No, well, I have an
Okay.
idea
Yeah.
about it. You know, when as it grows, it produces protons beyond, and when the protons get concentrated enough, it's difficult for it to grow. But if you've got a tube like that, the protons can not only grow this way, but be pushed off to the side. And so if you have something that's like penetrating through this sea of protons, the protons out here are not limiting it because they can go from one side or the other, and this can poke through the sea of protons. I think I'm not explaining it very well, but that's the general idea. And
Yeah.
interestingly, what we found is that if you shine light on this right along the middle of the tube, it moves away from the light.
Oh, interesting. Okay.
Yeah. So I think what's going on is that the EZ may be growing sideways, producing protons, and the protons are then pushing this structure away.
Got it. Okay. Interesting.
Yeah.
Very, very.
We should return to that issue or subject, but we haven't gotten to it.
Yeah. Awesome. So let's talk a little bit about. I have a couple questions about deuterium. I'm not an expert in deuterium at all, so I don't. All I really know is that essentially deuterium is, you can think of it as like heavy water.
Yeah.
Right. And as far as I'm aware, our mitochondria make water during cellular respiration, and that water is deuterium depleted water. So have you done any studies looking into deuterium or deuterium depleted water?
Yes.
Okay.
We just finished the study, and deuterium depleted water is supposed to be good for health. Mm-hmm. We studied deuterium depleted water compared to ordinary water, which contains deuterium, and we found that deuterium depleted water builds bigger EZs.
Okay.
Now, we haven't spoken about that, but in terms of cell function and health, your cells should be filled with EZ because it's the EZ transition, as you can read about in the book. The book was a little premature, but basically the general idea was the same, that there's a phase transition between the structured water and ordinary liquid water, and that transition is what basically mediates whatever the cell does, the action of the cell. Like in muscle, it would be contraction. In the nerve cell, it would be conduction. Secretory cell, it would be secretion. And then the water returns to the structured or EZ state. So it's really important to have a lot, or a cell basically filled with EZ water.
Yeah.
So if you have deuterium depleted water and it builds bigger EZs, then the cell's going to be healthier.
Yeah.
And that's what we found. So we have, the manuscript is written, and we're just about to submit it for publication.
Oh, awesome. I'm excited to read that. Could you explain a little bit about phase transitions? I've read more of the fourth phase water than I have cells, gels, and the engines of life. So I haven't. I don't think the complete best understanding of phase transition. So could you explain phase transitions, and also how does a cell know when to do these phase transitions to do work in the cell, if that makes sense?
Yeah, usually there's a stimulus of some sort,
Okay.
and the stimulus is what breaks down EZ water. Now, different cells have different stimuli, and you can imagine that there could be various stimuli that basically break down the EZ water. And if you break down the EZ water, it's going to convert back into ordinary liquid water. And have you heard of the action potential?
Yes. Of
Yeah.
course.
So action potential is said to be something completely distinct from the action of the cell. It's an electrical thing, but I think they're the same. So if the cell starts with negatively charged EZ water, stimulus comes, and the cell undergoes a phase transition, and the water goes from negatively charged EZ water to uncharged
Liquid water.
Liquid water. You'll get an electrical signal that goes from negative to zero.
Yeah.
Right. And that's what you get. That's the action potential. So I view the action potential
really
not
interesting. Wow. Okay.
well, yeah, it's not as a separate event. It's part and parcel of this phase transition that happens.
Okay.
Now, was there more? I don't remember the question exactly.
I guess. I mean, you kind of explained it. Like, how does a cell know when to do the phase transitions? But basically, some sort of stimulus occurs.
Cellular stimulus, and it could be any of a number of stimuli that basically cause the EZ water to transition.
Okay.
And then once it starts transitioning, it goes all the way.
Yeah. Do you have, or do you know of, or have you guys done any experimental evidence showing a phase transition going from EZ water to liquid water? Like, I know that occurs that occurs over time, right?
It should be quickly. It could be quickly.
Okay.
Yeah.
Is there evidence of it going quickly?
Well, if you take the action potential as evidence, the action potential is very sharp.
Yeah.
So it happens quickly. I think that you don't know about it, but the way the muscle contracts actually involves a phase transition. You know, we've studied the muscles for years, and I wrote a book in 1990 about this, and it involves phase transition. Although at the time we knew less about the phase transition, it involves the water. We have a paper published more recently than that, showing that the water actually, that the muscle undergoes, the proteins and water undergo phase transition as it starts contracting. So it's not exactly direct evidence, but it's evidence. So more evidence needs to be obtained for that, and maybe there is more evidence, but I'm blanking on it.
Awesome.
But you're asking questions that few interviewers ask.
So no worries. It's good to not know. I mean, we don't know a lot of things, so I'm okay with not knowing stuff. I want to well, I guess first, so you guys did a study where you put this sort of tube in water, and you saw a flow going through the tube.
Right.
Right. And then a later study, you looked at this, or I guess before you looked at that study and be like, this kind of resembles blood circulation within capillaries and with blood vessels. And then you did another study on chick embryos, kind of furthering that evidence, which seems very confirmed to me. But why don't can you talk about that a little bit?
Yeah, sure. So first to start with, it was a student like yourself, a freshman, I remember, and he joined the laboratory, and I gave him usually I offer like 2 or 3 different projects, and they get to choose which one they like to do. So this student chose a project. We had just found out that one of the polymers that we use very often in the lab is called Nafion. It's like Teflon, but it has charge groups. So it's hydrophilic. It produces beautiful exclusion zones. And we began using it. It came in sheets. We found out that it also came in tubes, 1 or 2 millimeters in diameter. And so the wife of one of my postdocs was working with that and said, hey, you guys should try. So I asked the student to see whether the curvature that you have in a tube would impair the development of exclusion zones. And apparently he got a result within a day or 2, and I had disappeared to Europe somewhere for a trip, and and he was bored. He was trying to figure out what should he do, and he was very conscientious, and so he started, and he found something that blew my mind again. That he put the tube horizontally oriented into the water, and there was a sprinkling of microspheres, so we could follow the water's movements. We can track it because the microspheres are suspended, and you can see them moving with the water. And he found that flow occurred through the tube indefinitely, just kept going and going. So first thing that happened, well, I should tell you the amusing, I was away in Europe, and at that time the use of email was, I think, not as common as it is today. And so I returned from my trip, and I hadn't heard from him. I didn't know what he was doing. And he confirmed a positive result. But I was sitting in my office, and I was sitting with a visitor, and I don't remember who the visitor was, but I think he was an important guy. But it was boring. it was not such interesting. And,
okay.
You know, so usually I leave my office door partly ajar because, you know, people who want to come in, they knock gently at the doors. Are you busy? You know, and I'd like to talk to various people, especially people in the lab. But this student was more brash than usual. So I'm sitting with this boring guy, hoping that he would leave or that all this would be over. And he kind of knocks gently, pushes open the door, and says, "I got something important to tell you. Is it okay?" So I pretended that I was irritated that, you know, he walked in so brashly, but I really wasn't, because I thought, well, maybe this could be the end of my conversation with this important guy. And he said, what I found after I confirmed that EZs do build next to the curved Nafion surface, I noticed that the flow occurred through the Nafion tube, and the tube was horizontal, immersed in the water, and it just kept flowing through. And I wanted to tell you about that. So, I was absolutely amazed by this because, you know, usually you need a pressure difference, hydrostatic pressure, to drive flow. Like in your cardiovascular system, the heart generates pressure, you know, and drives the flow. But there was none here, and I was thinking, well, we know that, or we knew that infrared energy was absorbed by the water, and it looked like his evidence suggested that infrared energy was getting transduced somehow into flow.
Yeah.
So we did follow-up experiments, and we confirmed basically everything. Well, it was, for me, it was a real revelation. Okay, so that,
I'm sure, yeah.
That's background. Now, I mean, imagine water as a transducer, right? You don't think of water as a transducer, but that's the way it seemed. Water as a transducer. And the energy looks like, yeah, infrared energy really plays a role. Yeah. So, okay, this is the end of the story.
yeah, but I just wanted to, but keep going.
Yeah, okay, so it was probably a few years later that I went to visit my friend in Russia, my friend Vladimir Vyakov, and he's a really smart guy and a good friend, and he's a professor of biology and the vice chair of the Department of Biology, which means he runs the department. And we're close colleagues, and we appreciate each other's work. So I go to Russia, and Vladimir entertains me at his home, which is an apartment right near Red Square, and we have caviar, which is very nice, and lots of vodka and such. Nice. You know, good friends, and the Russians, when visitors come, that's what they do. So he said, "I got to take you to the laboratory." So we go to the laboratory at Moscow University, which is right nearby, and it's their premier university, you know, comparable to like Harvard and Yale and such, and Davidson. So we go there. He said, "I want you to meet my colleague down the hall." So he calls the colleague. Colleague comes in, doesn't speak a word of English. So Vladimir translates, and the guy said, "There's a big problem in the cardiovascular system." Oh, so my eyebrows are up and my nose is in the air because
you're waiting to hear what
yeah,
is it?
I know something about the cardiovascular system. In fact, I did my PhD thesis.
Yeah. You modeled it?
I modeled it. Yeah. It was a time that I thought modeling was interesting. I think less so now because there are lots of assumptions that go into models.
Exactly.
Which gets me into trouble by criticizing. But at that time I was doing it, and I was modeling the pressures and flows in the systemic arterial system and the pulmonary arterial system. And I thought we had it all worked out.
Yeah.
What kind of major problem could there be? So I was behaving in a way that is, for me, unusually arrogant. Okay, come on, tell me. Yeah. Well, it took 5 minutes and he had to be convinced that there was a real problem. And I was kind of shocked. And the problem is not a complicated problem, it's a simple problem. So the problem is this. Red blood cells, which have to go through the capillaries, they're 6 or 7 micrometers in diameter. And the vessels, most of the vessels are smaller. They're 3 or 4 microns in diameter. So you can imagine, you got to push a red cell through the capillary. So what you need to do is you have to bend or squeeze the red blood cells. Yeah. And you can see videos showing that that's exactly what happens. They get squeezed. So this guy said, well, squeezing requires energy. How much energy is required? So he said to me, if the left ventricle, which produces pressure, if that pressure is sufficient to bend, squeeze those red blood cells, it would have to develop a pressure of extraordinary magnitude. He calculated it was either 1 million times or 1 billion. I think it was 1 million times what the pressure actually developed. So he said, it's impossible. Can't do it. There must be something else. And he starts giving me like a half dozen different hypotheses as to something involving bubbles in the system and a few other things. Okay. And meanwhile, I was thinking, we know that if we immerse a hydrophilic tube in water, the flow goes through, and it's run by infrared energy that comes in. And I'm thinking, boy, that's a really simpler way of achieving that. Yeah. So I approached one of my students, Jing Li, with, maybe you want to tackle this project and see if the phenomenon that we see in the lab is responsible for this. And he told me afterward, after he got a positive result, he told me, this was a few years later, he said, when you told me about this project, I was sure you were on some kind of drug because I can never imagine such a thing. Yeah,
it is astonishing. Yeah,
it's astonishing. Yeah, it's absolutely astonishing. So we agreed that he would pursue the subject, and he did, and he used the chick embryo, and chick embryo is useful because a 3-day-old chick embryo, it's the cardiovascular system is already developed. But the regulatory systems are not developed, so it's a pretty pure preparation. Yeah. And he was able to develop a way of imaging some of the vessels there. And he did the experiment. And the essence of the experiment is to see whether infrared energy drives the flow, because the phenomenon that we're dealing with, involves infrared energy. So the first thing he did was to stop the heart. So it means that the heart is not developing any pressure. If anything, it's an impediment because it's now a blob that's sitting in the way. And if you were to measure flow, it would be reduced by that obstacle that's in the way. And it turns out he did some research. ChatGPT didn't exist at the time, but he was able to do research to show that there were no fewer than a half dozen people who had done the experiment stopping the heart in a completely different systems, and they noticed that the flow continued. Oh wow! Didn't stop. Yeah. Lower velocity, but it continued. Yeah. And so that's the first thing he did, is stop the heart, and indeed he confirmed that the flow was continuing, low value, but remember, the heart is now an obstacle. Yeah. So we don't really know how much flow there would be without the heart. Yeah. And then he tested the signature feature our mechanism that you add infrared and that it increases. And I see diagram that you've got there. His
the
result.
diagram's amazing. Yeah.
Yeah.
If you want to describe the diagram for everyone.
Yeah. So so at first, when he stops the heart by injecting potassium chloride, flow goes down pretty far down, but not to zero.
Yeah.
And so it stays there for a while.
So wait, sorry. So this is when the heart is stopped.
Up at the top at time zero. It would be.
Oh yes. So got it.
Yeah.
So, got it. Okay.
Yeah. So the flow had been, substantial.
Yeah.
Then he stops the heart and the flow goes down, but it doesn't go to zero.
Gotcha.
It means something else is involved.
Yeah.
It goes to some fairly low level, and then after a while he turns on the infrared light, and it increases by a factor of like three times or so, and then it turns off the infrared light and it goes back down to baseline.
Yeah.
So
amazing.
Yeah. So it doesn't prove that our mechanism is the one that's at play. There has to be another mechanism, right? But it's consistent with the idea, and so what it means is that it's not, if our result is correct, I think it's correct, but it means that our cardiovascular system, it's not the heart alone that pumps the blood, but the vessels also act in pumping the blood.
Yeah, and this is also not like a new idea per se. I think like Rudolf Steiner, he proposed that the heart was not a pump.
Well,
that's dramatic, I know, and he also.
Yeah, I never understood that exactly, because you might be right, but if something develops pressure,
then
it seems it has to be a pump.
I agree. I agree.
So I think what he's was referring to, and other people have too, is that when the heart contracts, it generates a vortex, and that's true. But I don't think that denies the option that it's really a pump.
I agree, 100%. So perhaps I think a better conclusion would be the heart pumps blood, yet we have this other mechanism of action that helps to pump blood as well, and that would be this exclusion zone with the energy of infrared coming.
That's right.
Yeah, I think that's a good solid conclusion as of now with the evidence that we have.
Just one thing to add is that we don't know which one is actually the major contributor.
Yeah,
because you stop the heart, there's not much flow to start with, and as I explained, there's an amplification of the flow with infrared, but you've got this blob of a heart that's in the way. So any blood vessels that are pumping flow are going to encounter this obstacle, this blob sitting there, and reduce the amount of flow. So we're not sure of the magnitude that's contributed by the heart and the magnitude that's contributed by the vessels.
Yeah,
but in your cardiovascular system, the vessels are playing a role.
Yeah, amazing. I want to be conscious on our time. Okay, it's 1 o'clock, so that's probably around 2 hours.
Yeah.
I could sit here all day and talk to you. But do you mind if we go for just like maybe 20 more minutes?
Okay.
And then we can wrap it up?
Okay.
Awesome.
Sure.
Cool. I want to quickly wrap up and kind of bring this all back to health. I know you guys have done studies on certain things like turmeric and these compounds that are generally thought as healthy and healthful, and you've seen that exclusion zone increases with these compounds.
Yep.
And so that's definitely one aspect of how this all ties into our health. The infrared light, as we've talked about all throughout this podcast, we know being outside in nature has so many implications for our health. One of those being infrared light, building this exclusion zone, making our cells run how they should be ran. And so quickly, I just want to I want to quickly touch on this paper that you guys have done on Wi-Fi on EZ, and you guys have shown that Wi-Fi actually decreases EZ.
Well, how did you get hold of that paper?
I just looked it up. Yeah.
Because it's not published.
Okay.
I'm not sure. So there was a problem with that study, and the problem is a personnel problem. So the student who worked on that was very conscientious, came up with that result, and we went back and forth every week or so, and then he disappeared, and I have not been able to contact him.
Okay.
I don't know.
So maybe we don't put too much confidence in this.
Well,
perhaps.
when the person who did the study is not approachable or reachable, if anybody has questions, including myself, you begin to have some doubts. It might be perfectly okay, but it might not be.
Exactly.
And
good to know. Good to know.
Yeah.
Thank you for sharing that as well. And then I think let's just wrap up with practical takeaways for health. If you could just give a list that you think things that people can do to benefit their health from increasing their EZ.
Yeah, sure. Happy to do that. Well, the first thing to do is to drink a lot of water.
Okay. Because that water will then get converted.
Some of that water, some of it gets peed out or evaporated or whatever, but some of it gets converted into EZ water.
Yeah.
That's number one. Keep hydrated. As we get older, we get wrinkles and we're dehydrated, and we also tend to get sick. So, I tend to have problems more than you do, I presume, but we're dehydrated, so if you don't have enough EZ water, dehydration is essentially loss of EZ water. If you don't have enough EZ water in your cells, then this phase transition that I was talking about is not going to work as well.
Yeah.
And you're prone to get sick. So you really need to optimize the amount of EZ water. So it was a good question. So the first thing is drink water. The second thing is to do juicing. And so a lot of people know about it. A lot of people don't know about
My
it.
family and I, we've been doing that for far longer than I've even been alive. So,
is that right?
Yeah.
Wow. Wow. That's so interesting. Yeah, so you know how it works. It's you go to your backyard or front yard or whatever, and you pull off the leaves of freshly grown plants, and you squeeze the hell out of them, and you squeeze out the water. So what is the water? Well, the water is EZ water because you're squeezing the water from inside the plant cells, and the plants are freshly grown, so their cells are filled with EZ water, and you drink it, and it may not taste so good, but if you spike it with something that tastes a little bit better, you can drink it. And my late wife used to do it, go out in the garden, and we used to drink it, and maybe that's kept me healthy. But so you're basically drinking EZ water, and whether it's that EZ water itself that somehow works its way into your cells, or maybe it's the negative charge from that EZ water that permeates your body very quickly and converts any liquid water into EZ water, because we have evidence that that's the case. So, but it works, and I know a lot of health practitioners suggest that water as being it's the easiest thing you can do. I mean, water from the garden to preserve or improve your health. So that's another possibility. Okay, third possibility is go out in the sun. Infrared energy builds EZ water. You get a lot of infrared energy from the sun. The sun has been tainted as a villain from which you need to protect yourself, and it's great for the pharmaceutical business, exactly. Sun blockers, but that's not necessarily what we're talking about. So anyway, sunlight is good for you because the infrared energy builds EZ water. A fourth one is the sauna, or as the Finns say, sauna.
Yes.
It does pretty much the same thing as the sun. It's heat, and it's infrared energy that produces the heat. So you're exposing yourself to infrared energy, and builds EZ water, and that's why a lot of people, you go into the sauna or you have one using infrared lights, not maybe as good as the real thing, and you feel good afterward. And I think the reason you feel good is your whole body is being exposed to infrared energy. So wherever easy water needs to be built to give you a full complement, it gets built. So you go in feeling exhausted and you come out feeling revived.
Yeah.
Or you go in with a pain and the pain is greatly reduced. Yeah. You know, et cetera, et cetera.
I can attest to that. Again, my family, we have the same sauna that we've had longer than I've been even born.
Is that right?
Yeah, an infrared sauna. And you really do feel amazing afterwards. Yeah. And like whenever I'm sick, my mom says, go in the sauna, go in the sauna.
So your mom has great wisdom.
Exactly.
Mom wisdom.
Exactly.
She knows what she's talking about.
For sure.
Yeah. I remember also, it was a meeting in Finland, and above the Arctic Circle, and I was jet lagged. I gave my talk. We went to a kind of dinner, a reception, and it was getting to be 10 p.m. And all I wanted to do was to go back to the hotel, drive back in the bus, and go to sleep. And so the organizer of the conference, after the dinner, gets up and says, and I was sure he was saying, okay, we're going to get back on the bus. He said, now it's time for the sauna. And I said, oh shit, that's not what I want.
Yeah.
And
so I just want to go to bed.
Yeah, well, so I went in, I spent 20 minutes or so, and I came out, it felt like morning.
Amazing.
I had 8 hours sleep.
Yeah. Yeah.
So yeah, my own experience concurs, it really works. And I think the mechanism is just a buildup of easy water throughout every cell in your body.
Makes sense.
Okay, so that's 4. And then you mentioned the turmeric and such. And some of these herbs have been known to promote health since Ayurvedic times, like 5,000 years ago.
Yeah.
We've forgotten about it mostly, but right now people have kind of become interested in turmeric, at least. But there are a whole bunch of others. Basil is another one, they call it holy basil. Ghee is another, you know, clarified butter.
Yeah.
And we studied them, and all of them build easy water.
Yeah.
And so I think the reason that they're good for health is because they build easy water in your cells. It's straightforward. We actually tried also poison. We tried glyphosate, you know, the
weed killer.
The weed killer, Roundup. And at any and every concentration, we try to diminish the size of easy water. So I'm kind of wondering whether this is a general mechanism for poisons, and we haven't tested it yet. One of the reasons I don't like to expose the students to poisons, you know.
Very kind of you.
Well, I don't want to end the lives of those students prematurely, so we haven't studied much. But we also studied anesthetics, which, you know, diminish or depress function. They also diminish.
That was really interesting.
Yeah. So that's 5, and the 6th one. You asked for a half dozen, I think. There are more. I'm talking about the easy one. Is connecting yourself to the earth. Now, most of us have leather soles, and I think you do too.
Yeah, I think mine are like plastic at the bottom.
Oh, plastic. Yeah. So you're insulating yourself from the earth. Now, if you connect yourself electrically to the earth, studies have been done for quite a few years. It's good for health. And the question is, why is it good for health? And I've seen dozens of explanations. And I think one explanation for me is probably maybe the correct one, and that is the earth is negatively charged, and EZ is negatively charged. And if you, I mentioned moments ago, if you add negative charge to liquid water, it converts directly into EZ water. So if you take off your shoes and walk on wet grass, or immerse yourself in a mud bath, or even in the ocean,
go to the beach.
Yeah, the negative charge is seeping into your body, and wherever you need negative charge, or sorry, wherever there's not enough negative charge, which comes from the EZ, the electrons that are coming from the earth are going to fill those regions and restore EZ water. And that, I think, is a simple explanation for why earthing or grounding, depending on where you come from, is effective. So those are all kind of simple expedients and I remember, I know we need to end soon. I remember as a kid, I grew up in New York, in Brooklyn, and in the summertime, everybody went to the beach, and you could walk to the beach, and you couldn't get, you practically couldn't get from the street through the beach to the water because it was packed with people.
Oh, wow.
You had to step over people in order to do it.
Yeah.
So I remember once going with some friends of mine, and we were burying each other right near the water. And I was the last one to be buried, and I was buried up to here. And I remember so well the feeling I had. It was time to go. Everybody had to go back home, and the tide was coming in. You don't want to be beneath the water. And I just didn't want to get unburied. The feeling was like I'd never felt before in my life. Yeah. And I remember it to this day. Don't unbury me. and I was basically surrounded effectively by a connection to negative charge. And I think that's the explanation.
Yeah, so, I know James, you're familiar with James Oshman?
Yeah.
He's done amazing work on that, and Clint Ober, for people who want to look more into grounding. The science is there from what I've seen.
Yeah, the science is there.
Yeah, for sure. All right, I want to ask one quick question. Kangen Water. I have a Kangen machine at my house back at home. I think I've heard that you guys did some tests on Kangen Water, maybe?
Yeah.
Were there any conclusive results?
Yeah, the result was not conclusive.
Okay.
I also had a machine at home, and we have one in the laboratory. And I started drinking it myself.
Okay.
It's got high pH, and EZ has high pH or negative charge, and and I was thinking that it might be that there's EZ water. We tested it. We did not find EZ water, but I think the test was not conclusive.
Okay.
For technical reasons. We need to do the test again. But I came into contact with it when I communicated with a Japanese researcher and who was studying EZ water, or Kangen water, I should say, all his life. And he started telling me, he said that initially the results were so conclusive that the Japanese government decided that if anybody came to a clinic with a gastrointestinal issue, from your mouth to your anus, anywhere in between that, they paid for you to drink Kangen water. And I don't know if they paid for the machine or they paid for the water that was bottled, but they paid for it. And then he told me that there was some concern over the government was spending too much money, and did an extensive study, and they confirmed absolutely the same thing. And now they're paying for it not only for people who have gastrointestinal, people who have cancer also. The government pays for it, and so that's a good sign.
It's a medically grade water over in Japan, as far as I'm concerned.
Well, I'm not sure what you mean by medical grade.
I know they call it medical. I don't know.
Well, okay, sure. So I don't know why I stopped drinking it. Maybe because there were other waters, some spring waters that I started drinking, some of which have a lot of EZ water. Okay. But for the better part of my life, I was so healthy. I'm not going to be a champion basketball player, but almost never getting the flu or anything like that. Lately, as the wrinkles increase, some more things happen. But I don't know. Maybe it was the water that I was drinking for quite a few years.
Maybe. Well, I think you look very good as of now.
Well, thank you.
Well, Dr. Pollack, I appreciate this conversation so much. I'm sure the audience will get immense value from it. It was really amazing. So thank you so much for sitting down with me today. And
it's my pleasure.
Yeah, thank you so much.
Yeah, thank you for your time.
Awesome. Perfect.
Okay,
cool.
We've done it.
We've done it.
I got to tell you that I'm interviewed like at least once a week.
Yeah.
And I can't recall a more knowledgeable interviewer than you.
Oh, thank you so much.
You're amazing.
I really appreciate that.
Yeah,
I really appreciate
that. I didn't know what to expect. I figured nothing. And it was really amazing.
Thank you. Yeah. I appreciate it a lot. Yeah, I mean, this is my passion. So
when you graduate, come to work in my lab.
Hey, I'm ready. I'm ready. I would love to. I would really love to. Thank you for joining me in today's conversation with Dr. Gerald Pollack. If you'd like to explore more of Dr. Pollack's groundbreaking work, including his laboratory's latest findings and his insightful books, be sure to check out the show notes below for links and resources. If you're enjoying the podcast or learning something valuable from it, please support the show by subscribing to the YouTube channel. On Spotify and Apple Podcasts, you can leave a rating up to 5 stars and share a written review. This really helps others discover the show. Don't forget to check out the sponsor mentioned at the beginning of this episode. They really make these conversations possible. If you have questions for me, feedback about the podcast, or suggestions for guests or topics you'd like to hear about on the Whole Health Podcast, please leave a comment on YouTube or send me a direct message on Instagram. Once again, thank you for joining me in today's episode with Dr. Gerald Pollack.